and this is about GvHD prophylaxis, the novel strategies. And again, you know, there is no right or wrong answer. The idea is maybe we can start just by alphabetical order to ask what are the things that really impacted you in this field in the last 6 months or so. And if we go by alphabetical order, this is going to be Mutlu, I think, Arat. Sorry, Mutlu, it's not always good to be the first.
There are
Robert is smiling. He's going to be the last one.
Thank you. Actually, there are two aspects. So the novel agents are in our reach from our country on or not. We are aware about for the acute GvHD, the abatacept issue, and the other side, which are not available in our country. And we are not actually so now prone to use it on the front line. We are more or less still on our carcinoid inhibitor-based regimens, and the use of the future strategy in related methylated donors should we switch to post-infusion Cy or not as a T-cell depletion strategy. We are still waiting for the results of the comparative trials. We are not still convinced. There are many trials and actually many reports which are encouraging, but we are still as a center. But I know many centers in Turkey now switched nearly 2 or 3 years ago to post-infusion Cy in this mesh-related donor setting. In the MAS and the others, we are still sticking to ATG or post-infusion Cy. And there is, within the 6 months, I think we did not change. I think the level with the dual T-cell depletion issue, with the using of both ATG and then in a low dose and then going lower dose of cyclophosphamide is an issue in progress with now not 100 milligram, 80 milligram. So we are now switched to 80 milligram with the 50. Maybe in the elderly we did not use for anybody, but we are actually in the close follow-up all the data, and if we will have a comparative trial. And in summary, we are in this level in the GvHD prophylaxis. Thanks.
Superb. Thank you, Mutlu. Well, I said I'm going to go by alphabetical order, but of course, guys, please don't misunderstand me. You can jump in anytime you want. I can see Ali, who is very disciplined. He said I'm B, so I'll go second.
Good afternoon, everybody. So I will be trying to focus on 2 specific points. The first is GvHD prophylaxis in allotransplant following the use of checkpoint inhibitors. This is particularly relevant in some indications, for example, allotransplant for Hodgkin lymphoma. And in this setting, I think that 2 aspects are very important. The first is there should be a minimum delay of 6 to 8 weeks between the last dose of checkpoint inhibitor and performing the allotransplant. And the second is the mandatory use of post-transplant cyclophosphamide in that setting, not only when using a haploidentical donor, but also with other types of donors, such as much unrelated donors. And this has been shown in nice studies to decrease non-relapse mortality and to decrease GvHD incidence. The second is about the popular or controversial point of whether we should use in vivo T-cell depletion ATG or post-transplant cyclophosphamide, because the studies that showed the BMT-CTN studies were mostly in patients who did not receive ATG and showing superiority of PTCY over classical non-ATG containing GvHD prophylaxis regimen. And actually recent data from EBMT, rather than opposing these two strategies, is showing that the combination of ATG and PTCY results in a significant decrease in GvHD incidence without affecting the relapse incidence or leukemia-free survival. So rather than opposing these two strategies, I think that there is room of working on combinations of these two strategies in order to decrease GvHD, particularly when disease control is not negatively affected. Thank you.
Thank you, Ali. Ibn, do you share the same approach, or I guess things are slightly different on the other side of the Atlantic?
Yes, if you come across the pond, there is a little difference. So a couple of things from the standard of care perspective. As you all know, abatacept or abatacept was approved in prophylaxis for unrelated donor transplantation, but we at my center, we do not use it at all. And I'm not sure of any center that truly does use it a lot. Certainly the CTN 1703 results have changed our practice in reduced intensity, well-matched transplantation. That's predominantly now a PTCy-based regimen, and it's making its way into the myeloablative well-matched setting as well. It was already our standard for haploidentical and mismatched unrelated donors. So PTCy is definitely increasing in use. I will tell you that as we have done it, we are certainly not resting on our laurels there that PTCy is the end-all be-all. We have had bad outcomes with PTCy, especially in the older patients, some with borderline organ function, and there's a subset of patients that have really poor graft recovery afterwards that require a lot of help. And so we're trying to figure out, who should get PTCy and who shouldn't, or can we modify PTCy? And so for the older patients now, there's a subset that we reduce PTCy empirically, thinking it will modify the toxicities of such. So those are the standard of care changes. I think in the investigational realm, as most of you know, we presented the phase 3 data showing the addition of the vedolizumab was very helpful in the prevention of graft versus host disease, and that manuscript is in review at the moment, but it remains to be seen if and how that regulatory approval will be pursued, and if we're obviously still in discussions about that. The second avenue is 2 abstracts from us will be presented at ASH about the addition of ruxolitinib for GvHD prophylaxis to attack methotrexate backbone, showing very impressive results in the prevention of both acute and chronic GvHD. This will now become a phase 3 that we run to the CTN next year of comparing TAC methotrexate ruxolitinib versus PTCy in the reduced intensity setting. Here we've had discussions already that for the older patients, we may just switch to a TAC methotrexate Rux platform, given the really good outcomes that you'll see presented at ASH. And the goal of the phase 3 trial will be, I'm not sure we can show superiority, but it would be to show non-inferiority and provide an alternative regimen for older patients and those you feel who are too frail to receive PTCy, which we're increasingly running into. So those are the things we're...
Sorry, Ibn, did I get it well that you are running a randomized phase 3 trial now with TAC methotrexate and Ruxo, correct?
So it's TAC methotrexate Rux compared to standard PTCy TAC MMF. And that study is not yet open. It will be open next year. It is planned to be run through the BMT CTN in partnership with Insight. Yes. So that protocol is written. It's just going through the regulatory approvals and so forth.
Super. Thank you. So we are hearing about post-Cy. We're hearing now Ruxo. We just heard about also vedolizumab. So while we feel the case is closed, actually, it's extremely very active. We've been just joined by Amin Alousi. Hi, Amin. It should be early morning for you.
Hi there. Hi, everyone. Good to see everyone.
So we'll give you a couple of minutes to set. So where do I stand? Maybe, I don't know. Arnon, are you? Or at Attilio?
I don't know. I do not have a lot to add to my colleagues. We are using, as for us, we are using more PTCY. We don't do so much like in the States, mismatch unrelated by maybe on code based on the data that were presented lately, and maybe we should revisit it, but actually we are doing either a full match 10 out of 10 unrelated or haploidentical. I think that, and we're using it for haplo PTCY exclusively, and more PTCY also for the unrelated. I think that I agree that we need now to fine tune as for both ATG and PTCY, and the subset of patients that maybe benefit from one or the other, or like Ali, and you are doing combined. And there also may be different doses, different schedule that I'm not sure is optimized for at least PTCY and for the combination, and also for the side effect of infection, and so maybe we can optimize both for these heavily immunosuppressed patients. As for ASH, we repeated, of course, it's not randomized study, the CTN study, but comparing the exact group, but with ATG to the control arm. And here, this is a poster at ASH on behalf of the acute leukemia. And here we could not see any difference in GvHD, but the non-relapse mortality was lower with the PTCY arm. The end of the day included the other outcome, progression-free survival, overall survival, and GFS were exactly the same. And I would agree with Ali for the Hodgkin lymphoma. So this is the get checkpoint inhibitors with the data on increased GvHD. So for those patients, we are giving now PTCY. So this is another group of patients that we use PTCY in order to reduce the risk of GvHD post-checkpoint inhibitors. Thank you.
Thank you, Arnon. Appreciated. Attilio, so Italy is very famous for the haplo approach. So how do you handle the GvHD prophylaxis today?
30% of total allogeneic transplant procedure are currently performed by using post-transplant cyclophosphamide. In our center, we have the same experience with 25% of patients receiving this kind of prophylaxis. There are still some open questions about the dosage of post-transplant cyclo, because we have the knowledge that particularly in patients with elderly patients or patients with some comorbidities, full dose of cyclophosphamide are characterized by heavy toxicity, particularly cardiac toxicity. So we are exploring the new dosage of cyclophosphamide, 35 milligrams twice. Another aspect in this context is represented by the timing of calcineurin inhibitors, because, as you know, compared to the original schedule proposed by Baltimore, Italian experience evaluated the addition, the early addition of cyclosporine, and a retrospective AGMT survey showed that this early administration was characterized by a lower incidence of GvHD. But we have not solid data because this is a retrospective data.
How early?
Yes, this is
How early? Like day -2 or for the cyclosporin?
Yeah, early, starting from day +1 compared to day +5 after infusion. This was a retrospective comparison.
So just to, I mean, since we started using post-Cy 15 years ago, we always use cyclosporin at day -3, in contrast to whatever we've been told. And actually, we have data that it's slightly better than waiting, especially in terms of severe acute GvHD. So the concept of post-Cy wouldn't work if you start cyclosporin -2, -3, didn't prove in our hands to be true. But of course, we don't have any randomized studies. We have the registry, the EBMT registry data, which are totally in favor of day -2, -3.
Interesting. At MD Anderson, we've used post-Cy since 2009, 2010, almost exclusively, especially in the last, I would say, 8 years. So currently 95% of our patients receive post-Cy. Tacro and mycophenolate, both of those starting on day +5. We've shown, in comparison to our previous strategy, which used ATG for MUDs and TAC methotrexate for all, much lower rates of both the acute and chronic. Some of our analyses suggest even lower relapse. Not all subsets, but in certain subsets. The rates of grade 3 to 4 acute GvHD that we see with this is consistent with what's been published, and less than 10% and closer to 5% across the board. We do that in matched siblings, mismatch unrelateds, match unrelateds, as well as obviously haplos, and this has become our standard. I think it will be very hard to tease out when you have such low rates of grade 3 to 4 acute GvHD. Showing anything can lower it further would require such a huge amount of patients. So I'm curious to see the analyses about earlier starting of the calcineurin inhibitors. I believe it to be true, and I often questioned the long-held doctrine that don't give any steroids and don't give any calcineurin inhibitor till after the Cy. So I'm interested to see that data and that poster, and I'll be looking for it. You said at ASH from the European registry.
Yes, I mean, you mean the one with the ATG?
No, that one, I'm sorry. Yes, I'm interested to see that one as well, that comparison. But was there analysis that looked at, you said, within the registry, the European registry, looking at earlier use of calcineurin inhibitors in the setting of post-Cy? Is that good? Go ahead, please.
Yeah, it's from 2 or 3 years ago. First author, Annalisa Ruggieri. I can't tell you on the top of my head which journal this is, but either VMT or TCT.
Okay. That's interesting. I had not seen that, but I would like to.
I think it's, if I may, I think it's cancer, Professor Mohty.
Oh, cancer, maybe. Okay.
Cancer. We do increasingly in the last 2 or 3 years have looked at reducing the dose of Cy to limit the hemorrhagic cystitis, which we do see not infrequently. So maybe about a quarter of our patients develop grade 2 or 3 hemorrhagic cystitis, usually BK associated. And my sense is, and we haven't done the formal analysis yet, but from what I, don't take this to the bank, but what my sense is, there is a price with higher rates of acute GvHD, but also does seem to be better tolerated. So maybe in matched siblings, that strategy could be employed, but I think in higher risk donor types like mismatch or or haplo, I'm still a little skeptical of that approach. Even in the study that you guys are leading within the CTN, you use full dose Cy.
You mean the randomized study that's planned? It's full dose Cy 50-50. Yes.
Can you remind me the arms one more time? It's
sorry, I mean, for the sake of time, we have to move a little bit, because otherwise, I mean, this is fascinating. Robert, where do we stand? I mean, you have pioneered ruxolitinib for refractory acute GvHD, and now if we started using it for prophylaxis,
I'm happy that somebody is doing that, and it would be great if the results can be reproduced in a prospective randomized trial that even described. So I think it's great. I'm very happy that this happens, and I cross my fingers. I mean, in the end, we want to improve the outcome of our patients. If Ruxo can contribute to that, it would be great. Actually, in the initial mouse experiments, we gave Ruxo always prophylactically, not therapeutically. So it makes sense that it works also in the prophylactic setting. And I'm very happy to hear that. Here in Europe, there was not so much interest actually by the company to do such a randomized trial. So I'm happy that it happens now in the US. That's good news in my view. Since the initial question was what is new in GvHD prophylaxis, I think the vedolizumab data are really impressive in my view. I'm thankful that I could help a little bit with this trial that was mainly led by you, Mohamad, and you, Yibin. That's, in my view, the most exciting, besides what has been said already about PTCY. We're also using that in haplo and in mismatch unrelated donor, and sometimes also in match unrelated donors, not in siblings. So I think this is what is new in this area. That's already a lot in my view.
Well, superb. I think if I have to wrap it, and please correct me, it looks like post-Cy is really here to stay. Now we have a couple of question marks about the dosage, the optimal dosage of post-Cy, especially in the elderly and frail patient. And I must confess, we published our experience with lower doses. We have now 2 papers, 2 series from 2 independent centers going to the same results, showing that we can reduce by roughly 30% the dosage. The other thing that was mentioned by Dr. Bazarbachi initially, and there is also an EBMT registry-based paper about this, is maybe addition of small dose of ATG, like 2.5 milligram of thymoglobulin, may counterbalance what you alluded to, Amin, about maybe the more severe acute GvHD when you reduce the post-Cy dosage. We, of course, have the abatacept story, but I agree with Yibin that has been around this approval since 3-4 years, but we didn't see anything, I would say, rocketing in terms of using this drug. I may be wrong. The Ruxo study that was mentioned to be performed in a prospective fashion is an absolute delight for me. And hopefully we'll hear more about this.